Okay, everyone, let's get it started. I do want to cover very briefly one more topic before I move to color to get us ready for the final exam, okay, hopefully, to see a little bit more of all these different algorithms that we have discussed. And this is called image stitching. Now, how many of you have a cell phone that can do image stitching? All of you? Okay. So, for example, you can do a panoramic, right? Panorama, right? You put your cell phone here, you press a button, and you move, you rotate. Basically, it's a pure rotation, no translation, theoretically, right? And you create a sequence of images, and then there's an algorithm that stitches them together to give you a panorama, right? There are other algorithms that you may find in some online photo apps where you upload your photos, and it detects that you have uploaded photos of a scene from multiple views, right? But they overlap. And then it creates a larger view of the scene, right? You've seen this. How do they do it? What's that? There is template matching involved, but you actually need to understand the geometry of the scene and the camera motion, right, to be able to do this reconstruction. So let's think about this. And again, I'm just doing this because I don't usually cover this topic, but just to show you that all these things that you have out there on your cell phone, online, in all these apps, use the exact same equations that we have derived here. So imagine that we have certain number of images like this that we have discussed, and let's see how we can use the knowledge that we already have to define image stitching, okay? So say I have x1 through xn, my images, right? So these are my images. And obviously these are images of a common scene with some overlap, right? If there's no overlap, obviously there's no hope. So to see what we can do, and that could be similar to some type of homework that we may do or a question in an exam, right, that we have done. Let's see how we can model these things. So let's model, for example, the camera. I don't know why I have P as my camera model, but bear with my notation, right? So if P is my camera, that has my calibration matrix K, my rotation, and my translation matrix and vector. I can write this also in homogeneous coordinate system as K0, 0 vector, 1. And now here I'm going to have my rotation, my translation vector, a row vector of zeros, and a 1. And if I have, say, let my world point be, say, I call it PW tilde. It's equal to XW, YW, ZW1. Then the image point say, U, or why don't I call it X, I, or X, S for the image point. It's going to be my world point, right? Multiplied by my projection matrix, right? Here. Now, what do I have? I have my image, my first image, say image 1, or actually image 0, I call it. And this is looking at some scene here, maybe, right? So I have my nice mountains. I'm visiting Colorado. I'm in the Rockies. And I have this beautiful landscape. And I'm walking. I think this is beautiful. And I take a picture, right? And then I walk some more. And I take another picture. And for any point in my three-dimensional world, right, I get 2 image points, right here. I'm going to call x0 tilde and x1 tilde. And then I want to go from x0 to x1. And how can I do this? I need a matrix that maps from 0 to 1, right? Let's call it M10. And this is the matrix that does the transformation from here to here. So this is the matrix M10 that does that mapping, right? And what's this matrix equal to? Well, we know this. We know what this is equal to. This is equal to P1, P0, X0, right? In the same way, you could say, in the same way that I can compute this, I could also compute PW tilde, right, is equal to P tilde inverse times XS, right? It's the same equation, right? So I could go to P0. Now I could go to P inverse, that should be. And that will give me X1, right? So I could go from X0 to my original point here, right? PW, and then back to X1, right? What's the problem with this? So, I mean, this is the solution. This is the simple equation. The problem with this is I can't do it because I don't know the z value, right? The z value is unknown, right? Unfortunately. But we have defined the solution to this problem. Anyone knows what that solution is? So if instead of a point, I consider a set of points here, right, that are on the same plane, right? They have the same z, right? And I consider that point here. And now I compute the projection of this. And I get this point here and that point here. But I also have the corresponding planes here, right? And this, when this plane is at all the points on that plane are at the same distance. We have called this A. Anyone? The exam is coming up soon enough. Huh? You need to review these things. Homography. Right? This is a homography. So we know that we can solve this. For example, I could define that plane as n times p, all the points multiplied by this n vector, right? The normal vector plus some constant, right? So the normal vector plus the bias equals 0 would define this plane. And this is the homography I can compute. So I can say that x1 tilde is proportional to that homography from 0 to 1, right? So 1, 0 of x0 tilde, right? And that's what I want to find. And that's how these algorithms solve this problem, right? They can solve this by just applying a simple homography. And typically, if you want to implement this at home in MATLAB, typically the algorithms that you have on your phone use ransack to identify point correspondences, okay, with whatever algorithm of your choice for matching, right, for point matching, okay? And once you have identified your points, right, once I have my points, I solve with least squares. And where are the coordinates in the image for U1? We've derived this before. I'll write here a solution to that. This would be 0, 0. It's the 0th row, 0th column of H, right? We find the ij column and row of that matrix times U0 plus H01 V0 plus H02 divided by H20. You had this in the homework. That was one of the homeworks that you did early on this year. Plus H21 V0 plus 1. And plus 1 because we're assuming that all the z's are, right, at some constant. So remember that the H matrix defined up to a scale, so you have to define a scalar, and we're assuming the last component is equal to 1. And V1 is equal to H10 U0 plus 1 plus H11 V0 plus H12 H20 U0 plus H21 V0 plus 1, right? That's it. Simple as that. So it's basically a summary of the algorithm that you have in your phone or in any of these apps that do this reconstruction. There is a particular case to create these panoramas, for example, that is very useful, that we also could very easily understand, which is a simple rotation, right? Simple rotation. So the pinhole, say the elbow of my arm is a pinhole, right? So the elbow doesn't move, right? And only the normalized image plane, right? So I have a pure rotation. So I'll call these rotation panoramas. And this we can define as, let's see, U1, V1, 1 is going to be equal to F1, F1, 1 in the diagonal, 0 in the off diagonal, R10, it's a rotation matrix from here to here, right? And multiply by the focal length, the inverse of the focal length of the other camera, zeros in the off diagonal, times U0, V01. Or you could just simplify all of this into R10, U0, V0, F0, right? And now to solve for this, you need nonlinear least squares. And for that, you need an initial value for your homography, h. And now we need a function of h that allows us to follow the gradient, as always. And I'm going to define this function hw, w, the function that the parameter that I'm going to optimize, that is related to the rotation matrix, as follows. I'm going to define this as k1 times rw times the rotation from 1 to 0, k0 inverse, right? So if I take this out, this is a classical equation that we defined, right? And now I'm adding this additional term to modify the rotation using nonlinear least squares with gradient descent. And if I write this down as k1 rw, k1 inverse, k1 r10, k0 inverse, I didn't do anything. I just added this times this is the identity matrix, right? So I didn't do this. But now I'm going to add these two parentheses. I'm going to call this matrix D. And this is my homography from 1 to 0. Okay, you see that? This is the homography that moves me from one image to the other. And D is the matrix I need to optimize for. Now, D can be rewritten. This is the difficult step to know this, but you should be able to do this. K1 identity matrix times WX. Remember, this is the skew symmetric matrix that allows us to define the cross product. And this is the same then as 1 negative W2 F1 W, I'm sorry, Z, not W2, WZ times, F1 WY, WX1 negative F1 WX, WY F1 WX F1 and a 1, okay? So now you can do a one-to-one correspondence, and you can, with least squares, you can actually optimize for W, okay? And find the rotation. All right. This is you could see this is a quick overview of certain applications of everything that we've seen, right? And all the applications that come out of these things, these algorithms that we have derived here in class, are like this, right? They're just variations of the same thing. Now, doesn't mean that they are trivial variations, but they're variations of the same thing, okay? Right, any questions before I move on to color vision.
So do you
Yeah.
need to know the point correspondence before you do everything?
Before you do this,
Do you need to know point correspondence?
oh, for the rotation, you always do, right? For every problem in stitching, you always have to use RANSAC to find point correspondences.
Use RANSAC to find point correspondences?
Well, you need to use RANSAC. RANSAC is usually used to eliminate outliers. When you run any of these template matching algorithms, you're going to end up with lots of outliers, right? Because there are points that go occluded or change illumination dramatically or what have you. And there's just a way to eliminate. It's a simple way to eliminate outliers. All right, so let's move to color. Fascinating topic, very last topic of this class, this course. Hopefully it's going to take us only a couple of lectures to cover this. And then after that, we'll do a one review session before the exam, okay? Remember, the exam will be last day of the semester. Okay, so color. Why do we perceive in color? Anyone? Why do we have color vision? Make a guess. There are no wrong answers right now. It's better to make a mistake now than in the exam, right? So give it a shot. Why color vision? Why are animals colorful? I mean, look at birds, fish, even mammals, right? They all have a lot of different colorations in their skin. Why?
Well, to hide themselves more or try to scare their enemies.
Okay, so this is a very good hypothesis. So you may want to use some colors to avoid being prey by some other animals. What else?
To be attractive.
To be what?
Attractive.
To be attractive. Very good. Yeah, yeah, they're all right. I mean, they're all good hypotheses. But then, why do we have color vision? Say, we as humans, I mean, other than other animals. Why? Why have color vision?
It helps us survive.
Why does it help us survive? I mean, it's a good, you're not wrong, it does, but we could survive without it. Okay, let's think about this. Maybe I'll start with some slides maybe, and then I'll go back to the board. Let's do that. Think about what color is. Anyone can tell me what color is to begin with?
Just
Different frequencies of light. Okay. So here are some examples, right, of some birds. Look at the colors. It's outstanding, right? Why would nature do that? I mean, the short answer is we don't really know. There are a lot of hypotheses, you're right. Attract mates, for example. So there are, the coloration, especially in animals, tend to happen in males because males typically in nature have to convince females to mate with them. And females don't want to waste their resources with males that are not worthy of good genes. So it's a hypothesis, but that's the hypothesis. So the idea is that, you know, these colorations allow you to show to your mate, look how good my genes are and how great I am at providing to my offsprings, that I have enough energy, I eat so much, I have so much energy not only to survive, but also to create this extravaganza of colors and decorations on my body, right? This is how much extra energy I have. Which is all very good, except for the fact that it's known that in some birds, for example, when they are really desperate, they don't have enough food, or they're really sick and they're about to die, they will spend the males will spend the last amount of energy that they have to create these beautiful colorations so that they can mate and pass their genes before they die. So they lie, basically, right? We're not the only species that knows how to lie. So interesting. We don't really know, but they're very interesting hypotheses. But here's the first good news or bad news, depending on how you look at this. Color does not exist. The world doesn't have color. I'm sorry to disappoint you, but the world that you see around here doesn't have color, right? What we observe, it's an electromagnetic wave, right? Called light. Light is nothing else than an electromagnetic wave. That's it. And that electromagnetic wave has a variety of frequencies. And the frequencies that we can observe of that electromagnetic wave, it's called the visual spectrum. And it's this one shown here, right? But obviously, the electromagnetic wave may have other frequencies, right? So if you go here higher in the frequency, you have ultraviolet. You can actually see ultraviolet with ultraviolet sensors, right? And it's actually interesting. You can go online and just Google or go to YouTube or whatever your favorite search engine is and search for images or videos of cameras that have filmed in ultraviolet, and you'll see the world in another way, right? Using another part of the spectrum. Why don't we see that? We just don't, right? It's probably very difficult for nature to design these sensors, granted, but still, right? It is possible. If you go even further into the spectrum, you get X-rays, gamma rays, and so on. If you go in the other direction, you have infrared, right? So the one here in the visual spectrum that has the lowest frequency, or wavelength rather, it's red. And after that, we call it infrared, okay? And then we have microwave, what we use for radio waves, like FM, AM, and so on, long radio waves, and so on, right? Why do we only see this part of the visual spectrum? We don't know. That's why nature decided, right? Now the problem is that because we want to see this part of the visual spectrum, we want to be able to discriminate which part of the visual spectrum we're looking at. And the brain, very ingeniously, has come up with the idea of color. And it says, okay, from here to here, I'm going to call this blue, and from here to here, I'm going to call this green, and from here to here, I'm going to call this yellow, and so on, right? And in between, I'm going to call it different shades of blue, and green, and yellow, and so on and so forth. That's very smart, but that's just an illusion, right? The spectrum is a continuum. There is no such thing as red, green, blue, or anything else. And the world is not painted, right? What happens is that the Brdf function, right, the Brdf function that we discussed in shading, remember, observe specific frequencies of the light waves, right? So we talk about incidence, irradiance, and the reflectance of that, right? The radiance that pops out of the object. And that's given by the Brdf. Well, in fact, there's even more to that. More wave gets absorbed, or less of the wave gets absorbed, depending on the frequency, right? So if you see this red, that means that this object here has a Brdf that actually absorbs all the wavelength except red. So in fact, if color were to exist, this object would be anything but red. This is, so to speak, anti-red, right? That's why red pops out and you see it red, because the only thing that actually can escape that object, right? The only wavelength within the visual spectrum, that is. Right? I'll talk about this later on. So the question is, or actually, why don't I do this on the board first, and then I'll come back here. So where does this come from? People have been aware of color, or have studied color for centuries. And it was believed for a long time that white, the color white, was the purest color there is. Okay? And that different colors, red, blue, yellow, green, and so on, were constructed or were created, formed, when impurities were added to the white light. Which I know it sounds absurd, but that was the belief, right? So it was well known by the 1600s. It was well known that if I use a prism, and let's see, if I use a prism here, and I have white light, it should be white, not black, but you get the idea, right? White light coming in, then there is a spectrum that is going to be produced by this that goes, okay, I don't have all the colors, I don't have yellow, but from blues to greens to yellows and then to reds, right? And the idea was that the prism is adding impurities to the white light, and that's why you observe this. Then a famous scientist by the name of Isaac Newton, who you might have heard of, decided to think outside the box and asked the question, what happens if I now use a second prism here, right? If I add a reverse prism, so I now reverse this, and I put a prism here like this. What would happen to this? And what happens is obviously that these ones converge and white light comes out. Right? So I'm going from white to white, demonstrating that The prism is not adding any impurities, because if it were adding impurities, it wouldn't be generating white light at the end. So that instead what's happening is that white light doesn't exist at all, right? White light is a frequency or a combination of these frequencies. And what the prism is doing is separating the frequencies, the wavelength, right, into a rainbow of frequencies. And when you combine all the frequencies together, you're generating a single one, which looks white, right? Okay, and ever since the 1600s or early 1700s, when Newton proposed this idea, we see that was an experiment done in 1665. Ever since, we have known that that's what color, or what color really corresponds to. Now, color is very useful to us, and I'll talk briefly maybe about a few examples when I come back to the slides. Because we have this color vision, we associate different things to different colors, or different concepts rather, to different colors, right? And that's just because we learn to associate them, right? Because we perceive them to have that property. So, for example, you could also check that online. See if just look for an image of an apple, let's say, okay? Maybe a red apple, really nice, very delicious. And now look for a black apple, and I'm sure you can find an image online. And look whether it looks delicious or not. And most likely it's going to look really disgusting, right? But this is just someone that changed the color in the image. The apple is exactly the same thing, but you're going to associate it to something different, right? And that's because you have learned that this is not what an apple looks like, right? So I know you're too young for this, but maybe at least some of you will remember Coke and Pepsi had this idea a few years ago that was called, was it Clear Pepsi or Clear Coke, whatever it was? Crystal, okay. And it was transparent, right? It was colorless. And I mean, let's face it, Coke has this darkish color because there is a color added into the mixture, right? So they figured, why not make it transparent? And the idea was people will associate it to less calories, more healthy, right? It looks like water. The problem is that it looks like water. And people said that it was tasteless. It was very quickly discontinued. Complete fiasco, right? Because we associate transparency to water, and then when people spend dollars buying Coke, they don't want to buy water, they want a Coke. And Coke looks or has specific color, not water color. So complete fiasco. So these things are relevant. Also, when you design, and this is something you may work on someday, if you are to use what you have learned here to design signs or advertisements or what have you, color is a very important cue to use and manipulate, right? If you use color, two things can happen. So the way humans work, we have what's called a two system to process information, okay? System 1 and System 2, or slow and fast. There's this fascinating book I recommend that you read, written by a Nobel Prize winning psychologist who won the Nobel Prize for economics, because he demonstrated that we are not rational people when we make our economical decisions, okay? And he has this famous book, it's called Thinking Fast and Slow. Big book, easy to read, fun read, and you'll learn all about the fast and the slow systems of thinking. And system 1, which is associated to things that you automatically do as almost a reflex, right? You see A, you respond, right? Versus system 2, which requires reasoning about things, right? Now, if you want to build a successful ad campaign, you don't want people to use system 2. You want to use system 1. You want people to respond to that ad. And that's what advertisers do, right? If you design, say, traffic signs, right, for the road, you want signs to respond to system 1. You don't want people to have to reason about the sign. You want to say, oh, it's red. Red, it's danger. Therefore, I need to stop or need to be careful, right? Blue is information, and so on, right? This is the kind of association you want to build. You don't want to say, all of a sudden, we're going to use green from now on in our stop signs. Okay, what? It makes no sense whatsoever, right? So these things are very important to keep in mind when we talk about color. All right. Dimensions of color. Color can be defined according to its hue, saturation, and brightness. The hue is what basically you know as color, okay? Saturation is, if you will, the vividness of the color, okay? How vivid that color is. I guess people at the time of Newton would say the purity of that hue. So for example, a red velvet is of the same hue as cherry blossom. They have the exact same hue. But red velvet has a larger saturation, right? That's why it looks this red velvet strong color compared to cherry blossom. And then there is brightness, and brightness is nothing else than the intensity. Now, for many years after Newton, people wondered how colors are represented in the brain by humans. And by the late 1700s, early 1800s, scientists by the name of Thomas Young he was an English physician, speculated that the number of cones, right, or what we call now cones, remember the cones in the retina, the ones that are sensitive to different wavelengths compared to the rods that are only sensitive to light, no light, that the number of cones in the retina must be finite. And that is because a linear combination of a finite number of sensors will give you an infinite number of possible colors. And he reasoned correctly that this number had to be 3. And it is 3 indeed. The retina does have 4 different types of the human retina, that is. The human retina has 4 different types of cells, cones, and rods. Rods are only sensitive to white and black, and they see the whole visual spectrum, and it gives you the amount of intensity, right, of brightness that you have in that point, or at that point, this pixel. And rods are probably give you about this. This is the response of a rod. Now we have then 3 types of cones. They're called S cone, M cone or cones, and L cones for short, medium, and long wavelength. Now the short corresponds to approximately, not exactly, but close, approximately to the blue, what we would call blue hue, blue color, which is about here, which would be about, let's see, 443 nanometers. The L cone that corresponds to red is center about 574, and it's about here. And this is the L, right? So this is L, this is MS. And they correspond closely, not exactly, but they're almost blue-red. And the M that corresponds almost to green, that is somewhere here. And actually, it's closer. That's not correct. Something like this. And it's centered at 543. Okay. Now, if I, whoa, sorry guys. Wrong button. So if I go back to the slides, I can show you pictures of this. All right, so this is the color model that I have defined. Hue. In this model, you'll typically see these like 2 pyramids, one on top of the other, creating these 2 cones, right? Now, let's look at the center cone. This is connected to this. It's just been separated so you can see, okay? So if you go around the circle, this is hue, right? So you go from reds to yellows to greens to blues to velvets, back to reds, and so on, right? This is the hue. Now, as you go inside or out, you decrease or increase the saturation, right? And as you go up and down, you increase the brightness. So if you decrease the brightness, you go to black, pure black. If you increase the brightness, you go to pure white, right? And this cone is shown like this because this is what we can perceive. Our visual spectrum is this. Right? The brighter it is, the wider we perceive it, right? This is just our perception. This is not how the world is colored. It makes no sense that I increase the brightness and the hue changes from red to white. What? No, it's just that it looks white to us. That's our internal representation, right? That's how the brain interprets color. This is another way that you'll see this represented, but it just means the same thing. This is a representation that I've given you here. And you can see here better than blue would be probably this band right here, right? But that this is slightly shifted to a lower wavelength, right, than blue, but just by a tad. And red should be a little over here, but it's almost red, right? The long one. And the M, it's about green. Now, if you are colorblind, it does not mean that you don't see in color. It means that you may be missing one of these 3 different types of cones that there are in the retina. Okay? And depending on which one you're missing, you will see more colors or less colors. One thing that could happen is that, let me ask you this. For those of us that are not colorblind, or for all the people that are not colorblind, do you think that we all perceive the exact same colors? So even if we call them differently, right? Subjectively, we call them differently. Do you think that our perception of color is exactly the same, you and I? That this center right here is exactly the same for everyone, and that this variance of this function here is exactly the same for everyone? No, absolutely not, right? We all have a different experience. Now, depending on where this center is, you see how overlapped these 2 functions are? The M and L are superly overlapped. I mean, S, it's great, but M and L is like, gee, man, couldn't nature have given us slightly more separated colors, right? So that we could perceive more. So the problem is, in some people, they are so much overlap that they can no longer distinguish between greenish and red. And that's the most classical blindness, color blindness. People that don't distinguish green and red. Because it's not that they don't have these 2 sensors, they do. But the wavelengths that they observe are almost identical. Now, there is a solution to this, theoretically, or so the company claims. There is a pair of glasses that came out a few years ago that you put in, and they have a lens that separates the medium and long frequency. They separate it a little further apart, right? And for people that are colorblind for green and red, then all of a sudden they can actually make that distinction, right? And you can go online and see people that have bought these glasses for their kids or their spouses, and people start crying when they see color, and right? Like, well, okay. But it's a simple increase of that separation in the frequency, right? So you just make this this overlap a little less obvious, right? So that's a solution. But there are many different types of color blindness. Now, if you are not colorblind, you will see all the different types of color blindness that people might have. This is the original color, right? And then people may see different types of colors in the flag depending on the type of sensors that they are missing or the amount of overlap that you may have. Now, in the population in Northern Europe, or population of Northern Europe ancestry, as many as 8% of men, 8%, that's almost 1 every 10 people, are colorblind or have some type of color blindness, but only 0.5 percentage point of women. Why? What's going on here? And the reason is that the gene of color blindness is in the X chromosome. And lucky for you ladies, you have 2 X chromosomes. So if you happen to have a gene that is damaged, you have the other one that saves you. And the body is very intelligent. It will use the right gene, the good gene, to generate the right cones. But in males, if you're unlucky enough to inherit the gene that causes color blindness, then you don't have a second copy, right? Because the Y chromosome does not have that gene. And that's why there are many more cases of color blindness in men that in women. I mean, tremendously amount more. I'm sure when you were kids, you were all shown images like this. Now, this can go from really easy to really complicated, right? To really convoluted. And that this type of experiments will tell you whether you're colorblind or not. Everyone can see this one? Anyone can tell me a number? 12, good. How about this one? Okay. 5. They can get really difficult, I can tell you, because we are all colorblind, right? It depends at which degree, right? Because of these overlaps that I'm talking about, depends how much overlap you have. There's going to be a point where you can no longer make that distinction. All of us at some point. And remember, we only see a part of the visual spectrum to begin with. So obviously we are limited. So it's just the thing is that we're all different, right? And these are the different, this is a drawing of the cones that we have in the retina, right? And this would be the cone that responds to short frequencies, mid frequencies, and long frequencies right here. And then this data that you receive from these different cones is combined in the retina with these cells, typically bipolar cells are called, that combine this information. And I'll talk more on the board about this model, the opponent colors model. But this is what we believe is happening in human vision, that we create a model of colors that is represented here and called opponent mechanism. So I'll talk briefly about this. This is a representation of the back of the retina. Now remember that this is part of the fovea. Remember that the cones are almost exclusively in the fovea of your eye, right? So remember, if I could draw my cross-section of the eye, right? Terrible cross-section, I never know how to draw exactly, right? And here you have all the sensors, remember, that read the information that comes through the lens, right? Back here, that there is an area around here in the back of the eye that's called the fovea. And this is where we have most of the resolution, right? Most of the sensors. And this is the area that has the most of the cones, like 90% or 90+% of the cones. And this is a distribution of one subject, right? For red cones, blue cones, and green cones corresponding to long, mid, and short frequencies. You see those? Now, if I were to create this image of the distribution of cones for your retina and my retina, or your fovea and my fovea, they look different. So not only does our function, right, of each cone diverge, its center at the different position has a different variance, but also the distribution of the cones varies. So your experience of color, my experience of color are very different, right? There is a hypothesis that some women may actually have 4 different types of cones. And the reason is because if your 2 versions of the gene that generates this, that can produce or generate these cones in the retina are different enough, you may be able to actually end up with cones in the retina that perceive colors or frequency wavelengths at 4 different wavelengths, centered at 4 different wavelengths. There is anecdotal evidence to suggest that this is true. I'm not sure that this has ever been fully demonstrated, but it seems to be some evidence that that is the case. But in general, women are much better at color perception than men, meaning they have more discrimination than men do. So for the guys in the classroom, if you disagree with your girlfriend, I think she's probably right. Women have better color vision, just face it. All right, so before I introduce, let me chronodot. Before I introduce you to the opponent color model, I want to prove to you that this is a really good model. So I'm going to ask you to look at the center. There's a dot at the center of the next slide, okay? Look at that dot, and you have to stare at it for almost a couple minutes, okay? So be patient. I know it's exhausting, but be patient. Bear with me. I promise it's going to be good, okay? So look at the center dot, concentrate, don't move your eyes, and I'll let you know when it's over, okay? You ready? This white dot, just look at this. Don't move your eyes, just stare at it. It's not too long, you can do it. This is gonna blow out your mind. This is, again, how your brain plays games with you. And because of science, we know what it does, and we can actually play games with our brains. Keep staring. Don't move your eyes. I know it gets exhausting. It does. Be patient. There's a reward. First one that can tell me what happened gets a gold star toward the final exam. All right, 40 more seconds. You're almost there. Keep staring. Don't lose it. You're almost there. Keep looking. You're almost there. 15 seconds. Keep looking. Don't move. If you move, it's gone. All right, 5 more seconds. Hold it there. Keep looking. Don't move. What happened? Okay, gotcha. What did you see? How? How did you go from this to the American flag? Opponent colors. That's what happened. So let me tell you what happened. Oh no, I pressed it wrong. Ugh. All right. So what happens is I pressed the wrong button. We need to wait for like 30 seconds now, 40 seconds. So what happens is that, remember, your cells in your body, not just the retina, right? All the cells in your body stop responding after a while, right? They saturate and they stop responding. In fact, what happens here is that these cells start responding negatively. They are just so saturated after 2 minutes of staring at this, they start activating in the negative direction, right? So when I take this out, because it's trying to eliminate that information, right? It's like, this is bothering me. I want to get rid of this. I'm just tired of looking at this. I already know what this is. So it's trying to suppress the firing of these colors, right? And when it goes into the negative direction, it activates the opposite colors, what they're called the opponent colors to this. And what are the opponent colors? The colors of the American flag. And when I take this away, for just 1 second, right? It's very short, but for that 1 second, you have this beautiful perception of the American flag right there in front of you. It's like, and this is your neurons firing in the negative direction because they are suppressing that image, right? Okay, let's see. There we go. Okay, so these are called opponent colors. or opponent color theory, I should call it, but or color representation if you want. Usually it's called the opponent color theory. So what happens is that there's these ganglion cells that combine the cones. So you'll see cones typically represented in diagrams like this, with this rectangle that is truncated up here. So this is an S cone. I'm going to draw an M cone and an L cone. And what happens is that there is this cell in your retina that takes in this input from the S cone. So this is an excitation, so that activates the network. And then there is this one that deactivates the network, right, or the neuron, the cell. So this is inhibitory, and this is excitatory. Okay. And this is the output. So this is the first type. The second type is this one. Again, this is S, M, and L. And now I have the opposite. This and this are excitatory, and this one here is inhibitory. You see how this works? I have this bar here to indicate that inhibits, and this arrow to indicate that it excites, okay? So we can also say this is plus S minus ML, and this is plus ML minus S, right? And two more. We have this one, S, M, and L. This one does not use M at all, and it just has this inhibitory here and excitatory here. So this is plus L minus M. And final one, it does not use S. It only uses M and L. And it's inhibitory and excitatory. So it's plus M minus L response. So you see, what happened in this illusion that I just showed you is that there is these inhibitory cells, right, or inputs to the cell, the synapses, that inhibits the activation. So if I have too much activation here, I can inhibit this one, right? So I'm increasing the importance of this over time, of this input, and trying to decrease the importance of this one, right? And when I take this away, this is inhibiting so much that I create an illusion that this exists, right? When it actually doesn't. Now from here, we're going into two channels of opponent colors, and we don't really know where that occurs, but from here, this is combined into two channels. One is okay, there are actually three channels. Let me correct myself. There are actually three channels, but let me talk about the color channels first. One of them is what we'll call the red-green channel, and the other one is the blue-yellow channel, okay? Now there's another channel that also defines or that also comes from opponent colors, but that is not the color. This is the channel that defines shading. Why is that? The reason for this is that color, although we tend to think that color is three-dimensional, because I just said, right? The trichromatic representation is given by these three different cones. So therefore, color is three-dimensional. But in fact, color is not three-dimensional. It's two-dimensional. Because these three cones combined represent both color, that means hue, saturation, and brightness, meaning shading. Remember shading that we did, that we covered in detail? So shading is actually what defines this different, actually shading, let me say this, shading is what defines 3D shape. Okay? And shading is nothing else than if I have S, M, and L, right? My three different cones, I multiply this by, say, alpha S plus alpha M plus alpha L. And this combination, right, is the intensity of my pixel, or the radiance, if you prefer, the radiance of that I observe in my pixel, right, in my image, right? The amount of intensity I receive. It's a linear combination of these three different wavelengths. And the linear combination is given depending on the types of sensors that I have, right? The S, M, and L. Depending on what type of sensors you have in your retina, you're going to have a perception slightly different, or different values of alpha that are slightly different than mine, right? But the point is that this is what gives you what we would associate to a black and white image, right? That we only see grayscale. And that gives you the shading information. Now, color, or what we should call, because anyone remembers what we call this intensity? We had a name for this. Anyone remembers? Luminance, very good, excellent. So this is called luminance. So color is generally called isoluminant color. What isoluminant color means is that color, once luminance has been factored out of that representation, okay? Once I have eliminated luminance from that representation. So now let's think about this for a second. It's easier than it looks. And once you understand this is very important that you understand, because once you understand this, then you understand color. So imagine that you have an Rgb image, right? Rgb, as we'll see shortly, is a type of image that you take with a camera, right? Camera typically uses Rgb. Means it has a red sensor, a blue sensor, and a green sensor that respond to those frequencies that are centered at what we typically associate to red, green, and blue, right? All right, so imagine that I have an Rgb image. What does this mean? This is what it means. It means if I have that image, right, that I have taken, each pixel in that image, each of these pixels, right, is not just one value, but in fact, it's three different values, right? So it's like having a tensor here of three depth, right? Three matrices. And the first pixel gives you, so let's say x1 for the first pixel. It's giving you R1, G1, B1, right? The three entries of Rg and B for that pixel, right? So far, so good? Now, how are we going to represent that pixel? That pixel, I can represent it in RGB space, right? I can say, OK, this is my RGB space. And now, you know what? This RGB is going to have a value for R, a value for G, right? So I'm going to go here, and a value for B. So I'm going to go here. So this is my vector that represents my RGB coordinates, right? But this vector here, right? This vector, which is, we called it what? X1. This vector X1 has information of color and luminance, right? Now, what is luminance? Let's review this. Luminance is nothing else than a linear combination of my 3 channels, right? So in this case, because I have RGB instead of SML, I'd have that luminance is equal to alpha R plus alpha G plus alpha B, right? Right? OK. And what's this? What's this equation? Anyone can tell me what this is? So if this is, if that came from X1, what's this? What's that? Very good. This is a function of X1, which we called a norm, right? This is how I define my norm of my vector. Now, what is isoluminant? Isoluminant is x1 or xi in general divided by the norm of xi with the norm given by this function, right? So maybe I'll write f, this function here, right? Now, if all these numbers were ones, right? And I squared this and took the square root, that would be the Euclidean norm, for example, right? If it were the Euclidean norm, right? For a second, let's assume that this norm here, right? This norm of x is equal to the Euclidean norm, OK? Norm 2. Let's assume that for a second. It's not in general, but let's assume that it is. What would happen? This is what would happen. What would happen is that this norm, right, is what represents the luminance or the shading of my object, correct? And that means that if I have this vector here versus that vector here, right, I have a different amount of luminance. So that length, right, the norm of that vector, is what tells me the grayness of that pixel, right, the shading in that pixel. Now, if I eliminate that information by dividing by the norm, then I subtract this from the representation. What does this mean? If I were using the 2 norm, it would mean that, of course, these axes go to infinity in every direction, as you know, right? So it would mean that I'm mapping everything to S2, right? The 3D sphere, correct? I'm mapping every vector to have now, if I call this, say I call this xi hat. Now, what is the norm of xi hat? It's 1, right? Again, I'm assuming that this is now the 2 norm for a second, OK? Right? Which means that all the vectors would be on the surface of a hypersphere because all the vectors are of length 1. So it'd be a hypersphere of radius equal to 1, right? So all my vectors would be of length 1 and would be on this hypersphere. But what's the dimensionality of that hypersphere? 2, very good, right? It says 2, because the surface of the sphere, remember the projective space that went from that sphere to the plane, right? The projective space tells me that that sphere can be represented as a single plane, a 2D plane. There is a homology, right? A one-to-one representation, a one-to-one mapping from the 3D sphere to the 2D plane, right? From S2 to the 2D plane. This is a two-dimensional space. And what does this two-dimensional space represent? Color. When I move from one vector to another vector here in that sphere, what am I changing? I'm not changing the luminance. The luminance is the length of that vector. What I'm changing is the hue and the saturation, the color, right? So color is represented by this, is given by this 2D, or this sphere rather, or let's call it a two-dimensional sphere, okay? Or a sphere, the two-sphere, let's call it like this. Yeah, it's better, okay? In 3D space, but it's a two-sphere, it's two-dimensional. Okay, so far so good? Now, if I change the metric here, I don't use the 2 norm, but I use any other norm. This is still going to define the sphere in 3D space, a two-dimensional sphere in 3D space. Why? Because the norm defines all the points on the surface of a sphere that are at equal distance to the origin, right? That's a definition of a sphere. So yeah, that's a 2D sphere that we're used to. But you know what? If I change it to the 1 norm, I'm going to end up with another sphere, right? That looks different to me, but in the 1 norm, that's the sphere. All the points on the surface of that object are at the same distance to the origin, aka sphere, right? So that actually defines the color sphere, if you will, okay? And that is how you can change the color by just moving on that sphere here, okay? In that orton or quadrant of the space. And if you want to change the shading, then you move in that direction away from the origin, right? See that? Okay, so let me go back to the slides. Oh, am I past time? You never tell me, huh? Well, sometimes you do, but not when you're having fun. All right, so forget this. I'll continue this next time. All right, see you next week. Have a happy Thanksgiving.